As usual, I will start with a very quick summary of the company. So BioInvent, as most of you will know, is based in Lund, southern Sweden, roughly 100 people. And we are quite integrated company, which means we're not only doing the discovery of new mode of actions, so that means antibody and targets, but we also do our own manufacturing, including formulations, and of course then also clinical development. Currently, we're focusing on two promising clinical stage assets. Both are in phase 2. One is 1808 and the other one is 1206, and I will get back to more details later. They are very differentiated mode of actions in areas of high unmet need, and actually also multiple upcoming value inflection points. So basically what we have are two first-in-class assets that have shown activity in a number of different cancers, as I will show you. And also on top of that have a very good safety profile. So the technology, which I will not explain in more detail today, I have one slide where I just touch base on it briefly, but I just want to make the point here, has been validated through a number of collaborations with those pharma companies. And besides this, we have strategic partnerships with, just to mention a few here, MSD, Merck Sharp and Dohme, AstraZeneca, and CASI. We're well funded through a number of multiple upcoming near-term catalysts, as I also will show you later, and have the FDA backing. So we have fast track, we have orphan drug designations granted for both clinical programs. We're listed in Stockholm on the Stockholm OMX Stock Exchange, and our current cash, which is depicted here, gives us a runway into Q1 2027. So just a couple of words. So at BioInvent, actually, we are focusing on innovation. So as some of you might be aware, pharmaceutical pipelines are increasingly chasing the same targets. We are innovators. So we really discover and develop drugs against new targets. And we have focused our efforts on two specific targets. One is TNF receptor 2, and the other one is Fc gamma R2b. And those targets are now in phase 2, in various phase 2 programs in a broad range of tumor types. And 1206, which is targeting Fc gamma R2b, and 1808, which is targeting TNF receptor 2, are both being developed in hematological as well as in solid tumors, where we already have encouraging clinical data. Just very briefly on the platform that we have, this is a highly unique and diversified screening platform that we use. It's a reverse phenotypic screening, and I will not go through the various steps here, but what we can do with it, so we can basically discover the function and the efficacy first, and like this we can discover novel IO targets, as we have done for TNF receptor 2 and Fc gamma R2b, the two targets that I already mentioned on the other slide. But we are also able to identify uniquely functional epitopes on already validated targets, for instance, CTLA-4. So that is our current portfolio. 1808 is the antibody that targets TNF receptor 2. We run it as a single agent as well as in combination with Pembro, since we had strong preclinical data showing both single agent activity as well as good synergy with pembrolizumab. And we're currently pushing this in solid tumors as well as in T-cell lymphoma. Then 1206, we develop in non-Hodgkin lymphoma in combination with rituximab and acalabrutinib, and in solid tumors in combination with pembrolizumab. And also, as for the two programs or two arms that I just discussed on the TNF receptor 2 program, they are now phase 2. In non-Hodgkin lymphoma, we are also in phase 2, and in solid tumors, we just started actually first-line non-small cell lung cancer and uveal melanoma. Just a few comments regarding those names here. So whenever you see MSD, this is a supply, those are supply and collaboration agreements that we have here in these two cases. AstraZeneca, the same for acalabrutinib, MSD, of course, for Keytruda. And then we have partner 1206 for China, Hong Kong, Macau, and Taiwan with CASI Pharmaceuticals. So then going into more detail, and I will start with the anti-TNF receptor 2 program, 1808. And before I go into the clinical data, I just also wanted to make the commercial case. So it's actually significant unmet need, and that's why we can also expect significant market shares and economics. So at the beginning, we'll focus on CTCL, and you will later see the more details second line, which is roughly a 200 million, but then already we can move based on the data into first line, and this will be roughly a 500 million. And as you have seen, so we're now focusing also on ovarian cancer, first line, platinum resistant, and this is actually a very significant market of 1.5 billion. And then we feel that based on the data that we have seen as a single agent, but also the initial data that we've seen in combination, we can go for other solid tumors. So a very interesting market opportunity. The data releases for 1808 that are planned during 2026, we will have the phase 2a CTCL update for 1808 monotherapy and the combination with pembrolizumab already mid this year. And then during the second half, we'll have further phase 2a solid tumor data for 1808 and pembrolizumab in combination. And we had just a very interesting update just ahead of JP Morgan. So then going a little bit more into the positioning. So we feel that 1808 can be developed as a frontline for the treatment of CTCL. We have exceptional safety and tolerability profile. All available therapies so far have limitations in both safety and efficacy. And with a ORR of more than 40%, along with an excellent safety profile, we will have a firm position as a frontline treatment of choice. And that could be a potential market opportunity as first-line therapy. And the market opportunity is also achievable in the near term, as I will show you later. In solid tumors, you will remember that we have demonstrated single-agent activity in ovarian cancer, non-small cell lung cancer, and GIST, in addition, of course, to T-cell lymphoma. We also have demonstrated synergistic activity with anti-PD-1. That was the data set that we just released ahead of JP Morgan. And here also the very good safety profile that we have is ideal for combination in several tumor types. And as you know, when you deal especially with the more aggressive solid tumor types, it's all about combination. And then it's important that you have a good safety profile. So jumping then into the latest data that we have. So this is 1808 in combination with pembrolizumab, and that data was just released ahead of JP Morgan. So data cutoff was December 18. We could show a 25% ORR, 65% disease control out of 11 of 17 evaluable patients. So at 4 patients with partial response, 7 patients with stable disease. And also what I want to say here, so since this is immunotherapy, we normally see first a stable disease, and then over the scans, it develops into stronger responses and strong partial, or maybe at some time point even complete. The combination was very safe and well tolerated. And you have to compare the 25% ORR actually with pembrolizumab alone that has been tested, and that was just at 8% ORR. So that means we have a triple effect on the activity of pembrolizumab. So that means we have strong activity in both high-grade serous and clear cell ovarian cancer subtypes, which I think is great. And we'll have additional 20 patients that will be enrolled to validate and quantify the signal with an expected readout in the second half of 2026. So then this is just to remind you that we have shown single agent activity in ovarian cancer, in GIST, as well as in lung cancer, just to give you some examples. So this was the ovarian cancer, 63-year-old patients with all the previous lines, so end of the line patient, and this turned into a complete response after a couple of weeks, actually. The same for a GIST patient, in this case, a patient with 12 previous lines of therapy. So again, end of the line patient, we saw a very nice reduction, very strong partial response, and believe this has now turned into a complete response. And what you can see here is also the very nice activity. So basically what the antibody does is depletes the T regulatory cells and then generates an influx of CD8 positive cells. That means it changes the tumor microenvironment from tumor supportive into tumor hostile. So very nice. And the same we could show for non-small cell lung cancer. So now I'm switching to T-cell lymphoma, and this is maybe a little bit complicated plot, but this is basically the current landscape, and we are here. So this is 1808, and this is basically safety. So the more right you are, the more safer you are, and this is activity. So you can see that there's only one compound which is more active, which is this compound here, which is an ADC. but this is also very toxic. It has a black box label, and it's also very limited because it's only focused on CD30 positive patients, whereas we are much, much broader. So we also have a very good efficacy, but a much, much better safety profile. And this is, of course, single agent data. So that's the data that we have shown actually at ASH. Data cut off October 6, where we could show 46% ORR as a single agent, 92% disease control in 13 evaluable patients. You see the different patients, so Sézary syndrome, MF patients, one complete response. And this complete response we have now for almost 2 years, 5 partial response, and then a couple of stable diseases. And then we could also show response in a more severe form of a T-cell lymphoma, which is PTCL, where we had 1 partial response and 1 stable disease. So also it was very well tolerated and received orphan drug designation as well as fast track designation for this. And this is a couple of examples. So this is the PTCL patient. What you have to look for is those black areas here that are completely gone here. And here you can see, since we have a very strong skin involvement, that we can really improve those lesions in the CTCL patients very dramatically. The current plan is, so we have orphan drug designation, we have fast track designation, we have done the phase 2A, phase 2 part A. We're currently running the combination with pembro, see whether we can even increase over 46% ORR. Then we will start dose optimization. And then we start planning for potential pivotal study that could be started in 2027. So switching then for the remaining time to anti-Fc gamma RIIb, this is our 1206 antibody. Again, a very interesting market. So currently we're focusing first on follicular lymphoma second line, which is $500 million. And we have started first line in combination with pembrolizumab melanoma, and then we will also go for non-small cell lung cancer. So that has been started. So very significant commercial opportunities. And of course, if it works in non-small cell lung cancer in combination with pembro in first line, then we can also look for other solid cancer indications first line. So the data releases that will come for 1206 in 2026 is a clinical update for 1206 in the triplet non-Hodgkin lymphoma that will be by mid this year. And then the first phase 2A data for 1206 in combination with pembro for first line non-small cell lung cancer and first line uveal melanoma. So we have a very strong positioning in non-Hodgkin's lymphoma, especially in second line, in the combination with rituximab and acalabrutinib. We can, position us as a chemotherapy-free regimen. We are on track for an ORR of 75%. This antibody is a subcutaneous formulation, which improves, of course, convenience, and then it's very easy to combine with acalabrutinib and adds flexibility. Very good safety profile, no cytokine release syndrome, no neurotoxicity, which supports a broad use, including in community hospitals, because all our competitors, especially CAR-Ts as well as bispecifics, have problems with cytokine release, neurotoxicity, and increased risk of infection. In solid tumor, which of course, as I have shown you on the previous slide, is the largest commercial opportunity. The next trial is ongoing, is in first-line lung cancer. We enhanced the activity of pembrolizumab because we saw very strong synergistic activity in preclinical models. We have seen strong signals, as I will show you, in heavily pretreated patients already. That's why we're now going first line, and it's ideal for combination component with anti-PD-1, potentially in several tumor types. Going back to non-Hodgkin lymphoma, so this was the data that we have generated in combination with rituximab. Those are patients that do not respond anymore to anti-CD20 based therapy. And you can see there's a lot of green and a lot of dark green, which means a high number of complete responses, almost 60%. ORR of almost 60%, complete responses of 41%, and disease control rate of 86%. And this is very good. And those complete responses that we have that are high quality complete responses that basically last for a number of years. Then this is the triplet data that we have published at ASH last year. Data cutoff was December 1st, 2025. There we had ORR of 80%, complete response rate of 47%, which is very, very good. As you see, also a lot of green and dark green. And as I already said, the treatment was well tolerated, and the convenience and the safety profile of this combination positions us in a very highly competitive option in the evolving non-Hodgkin lymphoma landscape. The plans for the non-Hodgkin lymphoma, so we are basically by mid this year, we'll have finished the phase 2A signal seeking. So there will be an update by mid this year. Then we should have the full data set for the triplet. And then there will be a phase 2A follow-up. And then we start planning for a potential pivotal study that could be started, if everything goes well, already this year, but most likely next year. This we will go for a Type C meeting, agree the pivotal phase 2 design, and then go for breakthrough therapy designation, as we will do for the T-cell lymphoma with 1808. So then switching for the last 5 minutes to the solid cancer opportunity with 1206. So we have treated patients, end-of-the-line patients that did not respond anymore to anti-PD-1 or anti-PD-L1 therapy, and saw a couple of interesting responses in uveal melanoma and cutaneous melanoma. Just to give you the example, this, for instance, was a very strong partial response in a 69-year-old female with uveal melanoma. And that person has received actually a number of IO containing regimens and did not respond anymore. And then in the combination with 1206, we saw this very nice, strong partial response. We also have an example for cutaneous melanoma, where the person have received 3 lines of previous ICI therapy, with partial response as the best prior response to ipilimumab and nivolumab. And in combination with 1206 and pembro, we got a nice complete response. So we collected all those data, showed it to Merck, and basically suggested to Merck to do this. Basically, first-line non-small cell lung cancer and uveal melanoma. Merck has agreed to that, obviously, so have completed the escalation, and we are now running the signal seeking. So we'll have 30 patients in non-small cell lung cancer and 12 patients for uveal melanoma. And the next update, or the first readout for this, will be actually by second half this year already. So at the end, I would like to end up with the key catalysts and also mention we had a very news-rich 2025 second half. So we had the ASH data sets for 1808 in T-cell lymphoma and 1206 in non-Hodgkin lymphoma. Then just ahead of JP Morgan, we published the phase 2A data with pembrolizumab in ovarian cancer. And then by mid this year, we'll have further 1808 data in T-cell lymphoma, plus the full data set for 1206 and non-Hodgkin lymphoma. And second half of this year, we'll have additional combination data of 1808 in solid tumors with pembrolizumab. And then the first readout of the phase 2A data with pembro in first-line non-small cell lung cancer and uveal melanoma. Thank you very much.
Thank you very much, Martin. It's a very interesting presentation, as always. So you've made quite a thorough presentation of the pipeline, current research, and so on. And perhaps we could elaborate a little bit on the business side of things. So how would you describe the current state of business development discussions? Are you seeing an increased interest today compared to maybe 6 or 12 months ago for your technology?
Yeah, so basically, and I can first reply in general, and then I can talk a little bit about JP Morgan, where we just have been. But in general, I would say now we are obviously with phase 2A data or phase 2 data, where we can show efficacy, liquid as well as solid cancer. We are now spot on in the area where you can initiate discussions, and that's what we have done at JP Morgan. So we met with a couple of corporates that are very interested either in the hem-onc data or in the solid cancer data. So the release of the ovarian cancer data was very timely, just ahead of JP Morgan, and that was actually a trigger for quite a number of interesting discussions.
Awesome. Exciting. And have you seen any M&A activity in your field or any transactions that have a bearing on BioInvent?
Yeah, not directly, because first of all, as I mentioned already during my presentation, so most of the other companies, they're not really innovating, right? So they work on the same targets and then maybe have slightly differentiated compounds. So I think in that sense, BioInvent really sticks out, and that is maybe also some reason why the pharma companies or the corporates want to see more mature data, because we're working on targets that nobody else has worked on yet. But of course, once you can show the application and the potential, then of course the value might be much, much higher. So in that sense, there was no direct comparison yet. So of course, the field is focused currently on ADCs to a certain degree, bispecifics also still probably, and then of course radioligands. But I think around JP Morgan, I'm not aware, at least I can't recall at the moment anything at the moment that there was a bigger deal. I think it was actually at the end of last year where there were a couple of transactions that were quite interesting.
Right.
Yeah.
And that's a very big life science or healthcare conference, right? So
Yes.
did you pick up anything else that you found particularly interesting and perhaps had a bearing on BioInvent?
Yeah.
Market trends or investing in oncology or something?
Yeah. So JP Morgan, just for the audience, is the largest life science conference, you can say. And it's a couple of events. It always happens in San Francisco. And then basically all the life science investors and all the pharma companies and all the banks and all the analysts meet there, right? So that's kind of just for the background of the audience. And I would say the sentiment was actually quite good, much better than a year ago. So it's clearly positive. As I said, from the BioInvent perspective, we had a full schedule. So we started meetings on Monday, and we had our last meeting on Friday evening. So we had a full week of meetings. And of course, the main targets have been corporates as well as US investors.
Yeah. Well, it's nice to hear that the sentiment is growing. I mean, looking at BioInvent's share price is more or less, well, I guess, sort of in a sideways trend as of recently, but perhaps interest is picking up if risk appetite increases. Could you say a little bit about funding as well? I mean, your balance sheet as it is.
Yeah. So basically, as I said, so we have a runway until early next year. We have a runway into Q1 2027. And we are always exploring both. So obviously, the idea would be to get some non-dilutive funding into the company. But of course, we're also on the side always talking to investors that are potentially interested to support the company further. And I think based on where we are, because what I just said about corporates, this is also true for investors, because we are now in a space where it's very interesting, because we can say, as I said initially in my presentation, we have two first-in-class assets that are active in liquid as well as in solid cancer and have an excellent safety profile, which is quite unique.
We have a couple of questions from our listeners. One is regarding BI-1206 and the pembrolizumab in first-line non-small lung cell cancer. So, based on the biological rationale and the data generated so far, what are your realistic expectations for overall response rate in first-line non-small cell lung cancer?
Yeah, so what we are hoping that we see something like 50%. Right. But, this is, of course, speculative in the sense. So the only thing that we have at the moment is some preclinical validation. And then, of course, the first data that I have shown where we could show really nice responses in end of the line patients. But that's what we are hoping for, because and that's why we pushed also Merck into going to first line. We really would want to see a big effect. And if you have a big effect, then this data will be transformational for the company.
Great. And when is the next readout that would shed more light regarding this?
I mentioned that already. So the first readout will be second half this year already.
Second half this year. Another question is your pipeline beyond BI-1808 and BI-1206. So beyond this, which assets in the pipeline do you currently find most exciting from a strategic perspective?
Yeah, obviously, both our assets that we currently have paused, 1910, which is our second anti-TNF receptor 2, as well as 1607, which is the other version of anti-FcγRIIb, are interesting. And probably, if I have to make a choice, I would probably prioritize 1910. And they have been already tested in the clinic, phase 1 dose escalation, so they are more or less ready to go. And then, of course, and that's nothing that we share with the public, but Björn is very active with his team to identify new compounds, and there we also made good progress. And hopefully, at some time point this year, we could nominate the next candidate for clinical development there as well.
Right. And when talking about new candidates, could you say something about the range of assets you have in preclinical stage? How many currently are under development?
Yeah, so we always have a number, so couple. So let's say a handful of different assets that we are developing and comparing. And I think the pipeline there, as far as I can say, is very, very rich and very, very, very conservative to move something into clinical development, because then, of course, you start really paying money. But we could even consider at some time point to structure partnering around that, because there's also some interest with pharma in earlier stage programs.
Right.
So we don't have to develop all by ourselves. So there could be also, structure the deal around that and to collaborate that.
Mm-hmm. And the current runway, does that include any new developments, or is that just focused on the clinical portfolio?
This is currently focused on the clinical portfolio and the early stage programs as well, but not 1910 and 1607. So they're currently paused. And of course, our focus is currently on those 2 programs that I have mentioned here, plus the early stage programs that I mentioned that Björn is working on. They are also covered by this. Absolutely.
Right. Thank you very much for your time, Martin, and for a very interesting presentation. It's time to move on. If there was an audience in the room, we would give you an applause, but I will just have to say thank you. And of course, those of you looking online, you can always send a thumbs up or anything like that. So thank you, Martin.
Thank you, Lars.